Okay.
Okay, so yes, the family Nesticidae in Japan. I'm sure that most of you are familiar with this family. It's a rather small family, numbering around 280 species and 16 genera. Most of the components of this family are troglophilic, and we also have several troglomorphic species, meaning that they mainly live in caves or in cave-like environments, but some species also can be found in the litter of tropical forests. It's a quite widely distributed family. We can find it in all the continents. But if we check the distribution of most of the endemic species, we can see that they are grouped in some well-defined areas that we can call a kind of hotspot of diversity for this family. And among these hotspots, we can find the Appalachian Mountains, the Mediterranean area, the Caucasus and Carpathian Mountains, the southern China and northern Indochina, and also Japan. So actually, Japan is a kind of hotspot of diversity for this family. In fact, along the Japanese archipelago, we can find around 60 species or subspecies of Nesticidae and 5 genera. And this is around 21% of all the known species of this family in the world. So it's the diversity in the Japanese islands is a kind of large. And if you see the species, most of them are in fact endemic species. Around 95% of the species are endemic. And if we check the 5 genera that we can find in Japan, the largest one by number of species is definitely the genus Nesticus, which includes the 83% of all the species. And it's followed by the genus Cyclocarcina with 80%, which is morphologically similar, very similar to the genus Nesticus. So a good question is why this family is so diverse in Japan. And of course, this is a very difficult question to answer. So before I try to answering this question, we had another one. Is this diversity of the Japanese Nesticidae really correctly established? So these species are really good species, or maybe we are actually overestimating the number of the species in the Japanese islands. Or on the opposite, maybe we are underestimating them. We have still several new species that we can find. So to try to answer this question, we use an integrative approach. Of course, we collect new samples, and we try to study them using both the morphology and their DNA. And we decided to focus mainly on the genus Nesticus, since this is the largest genus of the family in Japan. And studying this species, we want to know if we could find also new species. We want to know the systematic position of these Japanese species in the family. And we want to know also additional information about their phylogeny, and in larger scale, maybe to start to study their biogeography and possibly their evolutionary history. So to do that, the first step is, of course, to have samples. So we started to collect as much species as possible all over the Japanese archipelago. And so far we collected 38 species, which is about 60% of the known species in Japan. And we recognized the species already described using morphology. Among them, we also found some species that we could not identify based on morphology, so we consider them like putative new species. But in the case of Nesticidae, sometimes there is a very high intraspecific diversity inside the same species. So we were not sure if the morphology can be really correctly separate and distinguish the species. To confirm that, we used the molecular analysis, in particular the DNA barcode, and we ran a species delimitation analysis. So we sequenced our samples, and based on the results of this analysis, we could see that the barcode confirmed the species which were identified by morphology. So the morphology and the barcode actually match each other's. We also checked the putative new species, and again, the species delimitation analysis confirmed them like separate species, so like new species. And we also checked some of the currently known subspecies, and the analysis showed that they are, in fact, good species. So not only subspecies, but they are actually real separate species. So the conclusion of this analysis was that the diversity is actually underestimated. So it's already pretty high, but it's probably far to be really defined. So it's probably underestimated, and new species can still be found. The next step was to reconstruct the phylogenetic tree of the Japanese Nestico species. So we used a multi-locus dataset, including 5 gene fragments, for a total of about 5,000 pair bases. And we included also some species from Korea that we found in GenBank, and also some representatives of the European genera and the other Asian genera. The result of this phylogeny is that the Japanese and also the Korean Nestico species actually merge each other's in a large and monophyletic clade, which is clearly separate from the other European genera, including the species Nesticus cellulanus, which is the nominal species of the genus Nesticus. So definitely the Japanese Nestico species actually are not Nesticus, but are a different genus. And if we include some species of the genus Cyclocarcina, we can see that they also merge together with the other Japanese Nesticus. So actually, the Japanese Nesticus are probably all Cyclocarcina. If we check more in detail the phylogeny of these species, we can see that the species actually merge in some subclades. In particular, we can identify 5 subclades, which are quite well supported by the phylogenetic analysis. And these subclades are also supported by morphology. We can find clear differences in the morphology of pulp and epigene of the components of each subclades. So 5 subclades were supported by both morphology and molecular. And if we see the distribution of these subclades along the Japanese islands, we can see that their distribution actually, in general, don't overlap, and they are distributed in different parts of the islands. For example, we have one monospecific clade, which is distributed only in the northernmost island of Hokkaido. The Korean species and the species from the southern island of Kyushu, they also form another clade. And we have a small clade distributed in the central part of Japan. And then we have the 2 largest clades, which are distributed in one in the central eastern Japan and one in the western Japan.
You have 2 minutes.
2 minutes, okay. I will go very quickly. And this distribution can give us some good information about a possible route of colonization of the Japanese islands. For example, one route from the north, which colonized the island of Hokkaido. Another route can be can involve the colonization of the southern part, and then other colonization routes or diversification for the other parts of Japan. So in general, this is of course difficult to study for now, but in general, we can see that their distribution of these clades is probably related with the geological history of the Japanese archipelago. So in general, the Japanese Nesticus can give us important information about the route of colonizations and pattern of diversification of the spider species in Japan in relation with the geological history of the archipelago. And for the future goals, of course, we want to describe the new species and maybe make new combinations, so moving the species to from Nesticus to the genus Cyclocarcina. And we want to explore more in detail the biogeography of these species and trying to time calibrate the phylogeny of the Japanese Nesticus species. And in a larger scale, we also want to explore the diversity of the other minor genera which are present in Japan, such as Nesticella and Speleoticus, since we also have some new species, including some which are clearly troglobionts, so with no eyes and living only in caves. But this is a work in progress and maybe a topic for a future talk. Thank you very much for your attention. I hope I'm inside the time.